Welcome to the 10th of the webinar series on genetics and evolution organized by the Journal of Genetics. I'm very happy to welcome Professor Henrique Teotonio. He works at the Institut de Biologie, Normale Supérieure, Paris. He obtained his PhD in 2000, working at the University of California, Irvine. He then did postdoctoral fellowships at University of Cambridge, Princeton University, University of Oregon. And in 2005, he became a group leader at Instituto Gulbenkian de Ciencia Oeris before moving to his present position at Paris. Professor Teotonio has been one of the pioneers of using the nematode C. elegans in experimental evolution work. His lab has created populations with alternative reproductive systems and standing genetic variation. They use these populations to address many important questions in evolutionary biology, including questions such as pleiotropy of gene function, effect of mutation, segregation, and recombination on associations among alleles between and within loci, and effects of inbreeding and assortative mating on the genetic architecture. Professor Teotonio.
Okay, so thanks very much for the invitation. It's a real pleasure to talk to you. And so today I'll be talking about adaptation to variable environments. And most of this work was done by Snigdha Day and Tiago Guzella, two postdocs in the lab, and also done in collaboration with Steve Prue from University of California in Santa Barbara. So when we talk about variable environments, there are two aspects that we should consider. One is that changes in the environment occur in the average of a given environmental variable, such as, for example, over here in the left plot, where we see that within the last 100 years or so, there has been an increase in the levels of carbon in the atmosphere. And for the future, there are predictions that the average levels of carbon will increase, at least in some of these models. The second aspect is that we should consider is changes in the variance as well of the environmental factor across years. Here on the right plot, we see that relative change in very wet days has also been increasing for the past 50 years or 100 years and is expected to increase in the future. So these levels of environmental fluctuation. So there are these two aspects to consider when we talk about variable environments. How can organisms deal with variable environments? Well, the first and perhaps easiest, or at least for some kinds of organisms to deal with, is just to migrate to more favorable habitats. The second option is to acclimate to these variable environments through phenotypic plasticity. And a third way, which will be the focus of my talk, will be to adapt to these variable environments, in particular through natural selection on standing genetic variation underlying gene expression. And I focus here on standing genetic variation because most organisms in nature will not have time to adapt through mutation accumulation given the time span of where these changes in the environment will occur. And so most of them will depend on standing genetic variation. There's simply not enough time for mutation accumulation. Now, how can we understand natural selection in variable environments? And evolutionary theory, the one that exists, indicates that when individuals are able to perceive information during development, their own development, about the environment that they will face at reproduction, then one expects that the evolution of phenotypic plasticity will underlie adaptation. When this information about environmental change can only be provided by the mothers, for example, in placental animals, then selection for maternal effects should underlie adaptation and population rescue from extinction. Of course, when information about the environmental change can only be provided by grandmothers or by grandgrandmothers, etc., so previous generations further down the path, then selection for transgenerational effects, then maternal effects should explain adaptation and population rescue from extinction. So today I will talk about 3 or 4 different stories that we've done in the past 5 to 10 years in the lab. One is about adaptation from standing genetic variation to average changes in the environment. The second part will be to explain how adaptation can occur by the evolution of maternal and transgenerational effects when population face changes in the environmental variance. And finally, briefly, I'll mention adaptation and the integration of multiple environmental cues. Now we do experiments and we use Caenorhabditis elegans, this roundworm, as a model organism. This is an androdioecious nematode, which means that natural populations we find hermaphrodites that are able of self-fertilization, but also males, and they eat bacteria, soil bacteria. Hermaphrodites, importantly, hermaphrodites can only mate with males, meaning that the frequency of males is actually just the proxy for the degree of outcrossing. And in terms of some of the genomics, they have 100 megabase genome size. They have 6 chromosomes, 1 sex chromosome, and they have about, in terms of diversity, in natural populations, about 1 single nucleotide polymorphism for about half kilobase, although it's highly heterogeneous across the genome. And what we've set up was to do experimental evolution from standing genetic variation with these worms. To do that, we crossed 16 isolates at high population sizes, so to form an ancestor hybrid population with variation from these 16 isolates. And then we maintained in very strict laboratory conditions this population. In particular, we maintained them in a 4-day discrete and non-overlapping generations with constant larval to adult density of 10,000 individuals. Importantly, we can deep freeze the worms and revive them at our will. And this is very important for evolutionary experiments because we can compare ancestral and derived populations for phenotypes at the same time in the so-called common garden assays, where we control the environmental conditions of several generations. And during the first 140 generations, we call this the domestication phase, we've done a lot of work on that. The most important points are that there was adaptation to the lab, but genetic diversity was maintained, and this cartoon over here shows genome sequences of many different lineages derived from the populations, but they are shown by each row, with each color representing the founder haplotype, one of those 16 haplotypes that we hybridized at the beginning. And as you can see, there's a lot of color, meaning that there is a lot of diversity, and the founder haplotypes were very well mixed. Okay. So the first story that I want to talk about is about adaptation to average changes in the environment. And if adaptation to future environment depends on de novo mutation, then it's intuitive to think that lower rates of environmental change will allow more time for the relevant variation to be generated. And indeed, several evolution experiments, starting from single clones and no sex during the experiments, confirm this expectation. So, for example, over here on the right plot, I'm showing that increased antibiotic concentrations during evolution of bacteria. You'll see that gradual changes or slower environmental changes allowed the survival of a higher number of populations than a sudden change in the antibiotic. However, as I said at the beginning, most populations in the future, even microbial populations, will likely depend on standing genetic diversity and less so on de novo mutation, simply because there is not enough time, given the generation time and population sizes, for new mutations, beneficial mutations, to appear. And so what happens when there is evolution from standing genetic variation under environmental change? So let's imagine that you can describe something called the fitness reaction norms, which is simply the relationship between fitness or fitness traits relative to the environmental value for each genotype, which are these lines, these colored lines over here. If we're trying to understand adaptation to an environmental value of 1, so an extreme case, then the time it takes the population to take there will affect its dynamics. In particular, if it's a sudden change, then we expect this gray genotype to be selected and to promote adaptation. However, most populations are finite, and in these circumstances, and in particular, if we do not have a sudden environmental change, if we have a gradual environmental change, there is opportunity to lose this gray genotype by genetic drift or by founder effect. In particular, first over here we have selection for this gray genotype, then for this blue genotype, and only later, if the gray genotype is present, will adaptation ensue. So we wanted to test this idea, and what we did was to perform experimental evolution to a novel, challenging condition, which was an ACL concentration in the medium. Everything else, in terms of demography, was the same. From starting experimental evolution from the ancestor domesticated population with standing genetic diversity. We've also manipulated the reproductive system such that reproduction occurred exclusive by selfing, and thus that there was very little recombination between extant genotypes during the experiment, and effectively we are modeling a multi-allelic locus to understand adaptation to changing environment. And so we did these experiments, and the dashed vertical lines indicate the sample points for individual genotyping, SNP genotyping, single nucleotide polymorphism, which we did at the whole genome level. And we developed a model based on the data during the experiment of these genotypes and fitness that we measured. We developed a model to infer the fitness reaction norms of standing genetic variation. We were able to identify more than 200 lineages, as well as predict the population genetic dynamics of all of these lineages given a given rate of environmental change. And here on the left plot, I'm showing just 2 of these lineages that were inferred, and the fitness reaction norms of these 2 lineages, or expected two lineages, as well on the right plots, the expected frequency dynamics of these two lineages. In particular, what we find is that under a sudden environmental change, we expect that this lineage called L28 would quickly sweep through the population, and by generation 100 or so would go into close to fixation. This same lineage in gradual populations, the middle plots over here, would only start to increase whenever these populations would face the high salt environment. Until then, there would be selection for another lineage called that we call L11, that would first increase in frequency, and only when populations faced high salt would start to decrease. Actually, this is the same lineage that we would find, or we predict to find, to sweep in the control populations, meaning that whatever this lineage is doing is independent of salt. And so we wanted to verify this model, and because we had genome sequences of many lineages derived from these populations, we were able to match the SNP data with the genome Illumina data, and to pick from the freezer the two lineages that we expected would explain most of these dynamics. And then we did a whole series of assays, essentially to confirm that our model indeed was accurate, in particular the fitness reaction norm inference. And as you can see over here on the right plot, we have under competition conditions, we have selection we have a measure of relative selection, and you see that the two lineages cross at around 225 of NaCl concentration, which is what was expected from the modeling. So this was a nice empirical verification of our modeling, but we have not addressed our original question, which is there a role for founder effect and genetic drift in the loss of the lineage, which we previously find is the best lineage, like the great genotype, during gradual evolution? Our modeling in particular was deterministic, so we could not predict the effects of founder of bottlenecks and genetic drift with our model. So to address this, we did another evolution experiment in which we took 7 populations that had undergone gradual evolution for 35 generations until they hit high salt population, and then we maintained them for 30 generations in high salt at large population sizes or at small population sizes. And again, we did genotyping, this time by pool genotyping a small number of SNPs to identify the lineage 28, which we know is the best lineage from standing genetic variation in a high salt. And we have many results. The main ones are summarized over here. From 3 of the ancestral populations, we find that none of the populations with further evolution in high salt led to the sweep or fixation of this best lineage, here shown by a cross. They all led to some other point for some other lineage which we cannot identify, but clearly they had lost this best lineage before, during gradual evolution. From these other 2 ancestors, what we find is, as expected, that, for example, the ancestral 4 population, we only see a sweep of the best lineage at high population sizes, while the 3 populations with small size don't show this sweep. As expected, that the frequency is relatively small, and so we were able to sample this best lineage only with high population sizes. The final ancestor shows that independently of population size, there was this sweep of the best lineage, meaning that in the ancestral population, the frequency of this best lineage was higher than in the other populations. So this shows that indeed adaptation is hindered under slower environmental change if populations depend on standing genetic variation, and we delimit this formally under 3 stages. In the first stage, when the environment is not so challenging, the best genotype in the extreme environment is actually deleterious, okay, under density independent conditions, and will be kept at lower frequencies than others, and thus could be lost by a genetic drift. At a second stage, when there is crossing of fitness reaction norms, there is reduced fitness variance, and thus, as a consequence, there will be reduced adaptive rates. There is a third phase, which we have some data indicating that it occurs, but that I have not shown you, in which the maintenance of other high-fit genotypes, like, for example, this blue genotype over here, could increase selective interference with the gray genotype, and this will occur particularly at high population sizes, and the best genotype, again, could be lost by genetic drift, despite being the best. Okay. So the population genetic dynamics are incredibly different from what we would expect if we just accumulated mutations de novo. Slower environmental change is actually worse for adaptation. Now let's switch gears a little bit and talk about adaptation to changes in the environmental variance, not changes in the average of the environment. So, as I mentioned previously, when individuals have information during development about the environment they will face at reproduction, then one expects that the evolution of phenotypic plasticity underlies adaptation. That's a little bit the story that I presented just before. But when this information about environmental change can only be provided by the mother, then selection for maternal effects should underlie adaptation. Now, me and Steve Plew did some theory on this to see when would we expect the selection for maternal effect. And we start by a definition of what is the environmental variance, and in particular the environmental autocorrelation between maternal and offspring generations. So here it's a cartoon of all the possible environmental sequences when we consider maternal and offspring generations. The x-axis shows the probability of transition between discrete environmental conditions 1 and 2, while the y-axis, it's the other way around, it shows the probability of transition between discrete environmental conditions 2 and 1. In the upper right corner, we see that this describes all the environmental sequences that regularly alternate between conditions 1 and 2. In the bottom left corner, we see that this space describes all the conditions that are regularly constant, mostly staying either in environment 1 or environment 2. And the diagonal over here shows when the environment is not predictable between maternal and offspring generations, meaning it's like flipping a coin, which we call irregularly alternating. Given genetic variation for maternal effects, we find in this study that there will be selection for maternal effects of the alternative phenotype when the environments regularly alternate, here in blue. That mothers will tend to pass on the same phenotype to their offspring if the environment is relatively constant, and that there will be a random phenotype when environments are irregularly alternating. A random phenotype is what's usually called bad hedging or risk aversion strategies, a phenotype that encompasses that. These blank spaces over here show that there are environmental sequences where there will not be evolution of maternal effects, and so just basically genetic determinism or phenotypic plasticity will evolve. So, do we know about maternal effects in worms? Yes, indeed, we do know about maternal effects. In particular, we know that when hermaphrodites are challenged with high NaCl concentrations since the larval stages, they will accumulate glycerol, in particular to survive osmotic stress. And as a metabolic trade-off, they cannot provision their babies, their broods, with sufficient glycogen, which is a sugar essential for the survival of the embryos in anoxia, so a lack of oxygen. So this is a maternal effect that we decided to leverage to do some experiments to test the theory. And in particular, we had these populations that I described before, in particular the gradual populations, which adapted to high salt already. Okay? And so we use these populations as the ancestor for new experiments. This ancestor, we see now that if we are trying to manipulate oxygen levels, in particular with oxygen or without oxygen in the hatching environment, the life cycle is composed of two generations, as shown over here on the left, and that fitness is defined as the growth rate over these two generations, depending on the maternal and offspring hatching environment. We find that the ancestral population, high salt adapted, indeed, that anoxia, when we apply anoxia to the offspring, the hatchability of the embryos is much reduced, and it's mostly independent of where their mothers were when they hatched, either in anoxia or normoxia. It doesn't make really a difference. Importantly, the mothers do not seem to suffer in terms of fecundity, whether if they have been exposed or not to oxygen during their hatching, their own hatching. So this is the setup of the experiment. And the design, what we did was for 60 generations, to regularly alternate normoxia and anoxia between generations, so that we could have the evolution of this maternal effect of the alternative phenotype, in particular, the maternal provisioning of glycogen whenever they face normoxia conditions, and their offspring will face anoxia conditions. After 60 generations, we assay them for growth rates, and what we find is, in particular, that whenever the offspring face anoxia, but their mothers have faced normoxia, there is a large increase in fitness, so they've adapted, while there is not much when the mothers have faced anoxia and they face normoxia. Actually, adaptation in fluctuating environments is measured by the geometric mean fitness of the environmental sequences that the populations have faced during their history, and we do find that there is an increase in this geometric mean fitness. The middle panels over here, the middle bars over here, represent environmental sequences that the populations have not faced during their history, and so any fitness response is a correlated response to whatever happened in normoxia and oxia sequences. Now, we see that there was evolution of maternal effects, in particular because the actionability response has increased when mothers face normoxia and the offspring face anoxia. Okay. Interestingly, this response comes at the expense of fitness, if you can say so, of the mothers. In particular, the fecundity of mothers when they've hatched in anoxia is greatly reduced. So you can have the evolution of maternal effects even at the cost of the mothers. And just to confirm that indeed there was the evolution of glycogen provisioning, we've measured using classical histological techniques, we've measured the amount of glycogen in oocytes, so prior to fertilization in the worms, and we indeed find that only when the mothers face normoxia will they increase the glycogen content because their offspring will face the challenging conditions. Now, to test for the evolution of bed hedging, we did also evolution in unpredictable environments in which the probability of changing environments between maternal and offspring generations was close to 50%, close to flipping a coin, also across 60 generations. And our results indicated that nothing much happened, or if anything happened, there was maladaptation, meaning that the worms became less fit. So there is no theory to explain this, in particular because we were able to maintain the populations during the experiment. And so what can explain this result? So there was no evolution of bed hedging. What can explain this apparent maladaptation? So in the similar way as we argue for the evolution of maternal effects, we can argue for the evolution of high-order transgenerational effects. In particular, when the information about environmental change can only be provided by grandmothers or grandmothers, etc., then selection for these transgenerational effects should explain adaptation and population rescue from extinction. Indeed, there is this information, even though we've constructed these sequences to be uninformative between maternal and offspring generations, there is information going back 3 generations or 4 generations, as can be seen over here by the autocorrelation of the environmental conditions of these sequences. What we did was actually to control the probability of changing environments between grandmothers and offspring, grand offspring, in 3 different parameter sets in which there was a negative autocorrelation, basically flipping a coin again, or a positive correlation between grandmothers and grand offspring environmental conditions. Do we expect the evolution of transgenerational effects of higher order than just maternal effects? What we collected data of growth rates, the per capita growth rate, which is slightly is a good surrogate for fitness during the entirety of the experiment. And what we ask is, can we a posteriori develop a model that explains these population dynamics better than, for example, a model only with maternal effects or only with phenotypic plasticity? And this was one of the models that we developed, in which the growth rate would be explained by an effect of anoxia in a given generation, the effect of normoxia, or a carryover effect over multiple generations, which in turn would be a function of what we call reprogramming which basically states the degree of heredity of this phenotype across multiple generations, and then a scaling effect. What we find is that this what we call carryover model across multiple generations indeed explains better the population dynamics than simply a maternal effect model or a phenotypic plasticity model. And this shows an example of the parameter that parameter values that we are able to fit. In particular, we find that there is an asymmetry of this carryover effect in permissive environments. So the norm oxia, here shown in red, we see that it takes about 3 generations of this carryover effect to be completely resetted back to baseline levels, while when individuals face the challenging environment, then it takes 6 generations to complete resetting. So meaning that it is the memory of some kind of memory is percolated down generations for a higher number of generations when individuals face the challenging environment. Okay, so the last bit of this talk, I would like to talk about auto and cross correlations between environmental variables. Up until now, we've been considering that both in the average and in the variance, there is only a single environmental variable. But of course, natural, as it's quite obvious, it's natural environments. There is a multitude of environmental variables, and it's unclear how organisms perceive and could transmit environmental change to future generations. In particular, there is an interesting question about that the environment that informs the individuals about the future and about the future change is not necessarily the environment where selection occurs. Okay, selection up until now in our experiments was an oxia. So can we disentangle the selective environment from an environment where the information is perceived and potentially transmitted independently of selection? And we, again, were lucky. From the literature, we realized that giving blue light to the worms, they are going to when we expose them to blue light, they are going to react, so they can perceive this blue light, but that the blue light exposure is not stressful. So over here, I'm showing you some results of growth rates when we expose an ancestral population, high salt adapted, to blue light in the dark bars or no light, so the ancestral condition, and you can see that growth rates are uniformly, independently of where mothers and grandmothers have hatched, will increase the growth rate of offspring when they are themselves grown in an oxia. So it's for some reason it's actually good for the worms to perceive the blue light. And so what we did was actually it's the same experiment, and the unpredictable environmental sequences. So when there is a zero correlation between maternal and offspring generations in an oxia, we've also controlled for whether or not there is a positive or a zero or a negative grand-maternal, grand-offspring autocorrelation, and we did all of these sequences. We imposed the blue light in a reliable fashion or in an unreliable fashion whenever we've given the blue light to mothers whenever the offspring would face the challenging anoxia conditions. Okay? And so the idea is to see whether or not we have the evolution of a blue light dependent response, and if that evolution is dependent on the degree of autocorrelation between grandmothers and grand offspring. And the preliminary data indeed indicates that there is adaptation by the integration of multiple environmental cues. In particular, in the presence of light, when we compare the light worms grown in anoxia, so the offspring grown in anoxia, with light given to the mothers or no light, we find that reliable populations, here shown in these blues and these rectangles, seem to have evolved a positive fitness response, but not when they are not given light, while the unreliable, so the ones that have absolutely no information about the future environments given by the light, have actually gone down in fitness, but that this depends on whether the grandmothers or the mothers were exposed to anoxia. Okay. We further think that this response depends on the grandmother-grand-offspring autocorrelation during evolution. In particular, if there is a positive autocorrelation, so grandmothers face anoxia, then they will probably prepare their daughters, the mothers, to receive the blue light cue in order for the mothers to provision the offspring with glycogen for their survival in anoxia. So in other words, even though this is very complex, we were able to separate the effects of environment, of perceiving the environment from the environment where selection occurs. Okay, so in summary, adaptation to slower environmental changes might be hindered if responses only depend on standing genetic variation. We've seen that when individuals cannot access information about the environment they will face at time of reproduction, then transgenerational effects are expected to be selected depending on the degree of environmental autocorrelation across generations. Maternal provisioning underlies adaptation to correlated fluctuating environments. Carryover effects over multiple generations can increase population viability, particularly when there is little information about the selective environments. And finally, and more mechanistically, it seems to me that there could be asymmetric germline reprogramming, and that this could be adaptive in the sense that individuals keep a memory of the experience of past generations in stressful environments that may be necessary for their immediate survival in that same stressful environment, but less so when the environments are permissive. And I would like to finish by acknowledging Snigdha Dipde. He was really the spearheaded most of this work, and unfortunately and sadly, very sadly, he passed away when he was in the middle of trying to complete some of these experiments. And also, I'd like to acknowledge the lab support and funding for this work, for supporting this work. And thank you, and I guess I'll take some questions now.
Thank you, Professor Teotonio, for a very nice talk. So first question is from Amitabh Joshi, and this is his question. If you think back to your PLOS Biology 2016 work, is it true to conclude that the mothers in this work are plastically responding to generate the appropriate maternal effect?
Yes, I do think that that's the case. except that for fecundity, I'm not entirely sure the mothers will have less, themselves will have less glycerol, presumably, when they are faced with anoxia. But we haven't measured glycerol. So the question over here of plasticity is more, the way I see it is that for a given generation, you do have what's usually termed phenotypic plasticity. For maternal effects, it's essentially the maternal environment that will determine the offspring survival. And I believe that fitness in these circumstances is literally just can only be defined relative to a 2-generation cycle, not a single generation cycle. So I'm not sure if that was clear, Amitabh, but.
So he has a follow-up question. This is about the modeling work. How do you rule out the possibility that adding carryover terms or extra parameters to the model gives better fit just because there are additional parameters rather than a real biological effect?
Yeah, we cannot. I mean, that's the whole problem of model fitting. So, the story behind it, when SNCC started to work with me, and because of you, Amitabh, I believe, he wanted to measure these growth rates as during the experiments, and I said, yeah, but I don't know what we can do with them. And then we were able to do these ad hoc models. But yes, until we find a mechanism, we cannot. Yeah, you're right.
Okay. Uddalok Bangabash has a question. His question is, in the experiments where you measured the L28 and L11 fitness norms, were the cultures composed of only one genotype or were there multiple genotypes?
I've only shown with those 2 genotypes competing side by side at 50%, but we also have just single cultures, and the results are exactly the same. So it does not seem that there is frequency dependence.
Okay.
But importantly, during the assays, those frequencies between those 2 genotypes were never realized are not the same as during experimental evolution. Okay, so we cannot absolutely demonstrate that there was no frequency dependence between these 2 genotypes or with other segregating genotypes that we have not measured.
Okay. Professor Ranganath, you have a question?
Yeah.
Yeah, please.
Excellent piece of question and excellent piece of work, sir. And are we discussing something related to epigenome in evolution? Grandmother to X?
If I didn't understand the question. If that is if what I call carryover effects are epigenetic effects?
Yeah.
Yes. Yes. I mean, classically, epigenetics is anything. It simply means over the genetics. It simply means gene expression. It means development and physiology.
Yeah.
In modern times, epigenetics has been defined as things that pass on, some phenotypes that pass on between generations. I prefer to call it transgenerational effects or carryover effects. But yes, and in C. elegans, we do know quite a bit of what it could be. It's either RNA-based, meaning that RNA is passed in the germline, or, and perhaps more likely, the methylation patterns of histones that pass through the germline. Like mitochondria, probably not. Yes.
So can I say, I'm not sure, it is an extended phenotype of the grandmother?
Yeah, exactly. Yeah. You can think about it that way as well, which is the extended phenotype.
Thank you, sir.
Yeah.
So there's one more comment from Amitabh and a question, and I think we all agree with the comment. He says, very interesting work, Henrique.
Thanks.
He also says, would you care to speculate whether the memory of stressful environment mechanism might be very dependent on the type of organism? For example, worms versus insects.
Yeah, I'm not sure. I think that I don't know. So I can tell you two things, a couple of things. So usually, one is about bed hedging. So bed hedging in the literature, it's usually there are very few examples, even though many people claim about bed hedging, that the mothers can diversify the phenotypes of their offspring so that they can survive challenging environments. And it's unclear that even in the best examples, such as plants like seed banks, that's the individuals themselves do not have information or are not able to perceive information about their future environment. So I think that's something that our work shows, is that probably bed hedging is not as common as people have claimed. Then on a slightly different take on it, I think the reproduction system will have a major impact on how these carryover effects could underlie adaptation. In particular, all of our experiments were done under exclusive selfing. Okay, meaning that there is little opportunity, and since it's from standing genetic variation, it's basically just a big competition between extant genotypes. There is no novel recombinants, and there is little opportunity for parent-offspring conflict and little opportunity for sexual conflict. So in fruit flies, for example, you have males and females, and you have standing genetic variation. So you have both of these processes could lead to different dynamics, adaptive dynamics, simply because maternal effects are not perhaps sufficient to promote survival of the offspring, and there will be like sexual conflicts will take primacy. So, for example, placental animals like us, okay, we think that yes, there is maternal effects, and there are maternal effects, and we usually think that they are beneficial to the offspring. But we also know that there are many things that they are deleterious to the mother that the offspring do. And what we believe, I think that people believe that was the actual adaptive value of being placental was that all of a sudden mothers or females had to choose very carefully the males with which they would mate, otherwise their babies would probably not be very good. So it has nothing to do with the possibility that environment fluctuates, that you have maternal environments. It's simply a change in sexual selection. Okay, and we are seeing the consequences of that. So I'm not sure, Amitabh, if that's enough speculation for you.
Yeah. I guess we have answered all the questions. I guess there are no more questions. Since there are no more questions, yeah, please go ahead.
No, thank you. Thank you for hosting me and for the questions as well.
Thank you so much for agreeing to give this talk and for giving this talk, and it was a fantastic talk. Very nice set of work. Thank you so much.